Hi everyone, and welcome to our video on enzyme inhibitors. So this is our 7th video in our enzyme series, and what we're going to be doing in this one is having a look at the specification reference 2.1.4f, effective inhibitors on the rate of enzyme controlled reactions, which will look at both competitive and non-competitive inhibition. The first thing that we need to know then is what we mean by this word inhibitor. When we talk about an inhibitor, this is just a substance that either reduces or stops a reaction. And the way that that comes around is by affecting how the substrate binds to the enzyme, or it can alter the turnover number of the enzyme. Let's start off then with competitive inhibition. Now, competitive inhibition requires a competitive inhibitor. And the key thing about these is that they've got a similar shape to the substrate. So because our competitive inhibitor has that similar shape to the substrate, they would both fit into the same active site. So basically what we find here is competitive because the inhibitor and the substrate are going to be in competition for the enzyme's active site. If the inhibitor binds, then the substrate can't bind. If we then start off by having a little look at the diagram on the left that I've drawn with my amazing skills in Microsoft Paint, then what we can actually see is we've got our lilac-y thing, which is going to be our enzyme, and of course our enzyme has its active site. Now the active site of course has this very specific shape which is complementary to our substrate molecule, but in this case it's also complementary, at least in part, to the inhibitor. So what that means is if the inhibitor collides with our enzyme, then what's going to happen is we're going to form an enzyme inhibitor complex like we've got on the right hand side here. Once the inhibitor is bound to the active site of the enzyme, it prevents the substrate from doing the same. So basically, for the more enzyme inhibitor complexes we form, there's a reduced number of enzyme substrate complexes that could form. It is worth bearing in mind though that many of these competitive inhibitors are binding reversibly to the enzyme. So that means that they will be released at some point if the conditions are right. If we do have an inhibitor that binds irreversibly to the active site, we change its name ever so slightly and we refer to it as an inactivator rather than an inhibitor. Because once we've bound irreversibly to the active site, we have inactivated that enzyme permanently. If we have a little look at the typical graph that we would get, so we've got substrate concentration on our x-axis, rate of reaction on the y-axis. So what we can see first of all is with the green line, the one closer to the left there, that's our reaction line without the inhibitor. So that would be just our enzyme and our substrate together. When we add a competitive inhibitor, then what we can see is that we do get this reduction in the rate of reaction because the red line is lower than obviously the green one. However, if you look at this end of the graph, what we can see is there comes a point when they're both at the maximum rate of reaction. In terms of why that is, what we're looking at here is increasing substrate concentration. So if we increase the concentration of substrate, what we're then doing is giving a greater probability that the collision that occurs with the enzyme is going to be with the substrate rather than the inhibitor. So eventually, once we've increased the substrate concentration far enough, then it's just likelihood that it will be a substrate and enzyme that collide rather than the inhibitor and the enzyme. So we end up reaching that same maximum rate of reaction eventually. The second type of inhibitor we need to know about is the non-competitive inhibitor, or the process of non-competitive inhibition. The key thing about this is that the inhibitor is not binding to the active site. It's going to bind to a different region of the enzyme called the allosteric site. So if we have a look at our little diagram in the bottom left, we've got our enzyme again, we've got the active site. That active site is complementary in its shape to the substrate molecule, and then we've got this little region at the bottom, which is our allosteric site. Now, the allosteric site is where our non-competitive inhibitor will bind. So when that non-competitive inhibitor binds to the allosteric site, it's actually going to cause disruption to the tertiary structure of the enzyme itself. When we disrupt the tertiary structure, we're changing that three-dimensional shape, and because of that, we're changing the shape of our active site. So what we find is there's this slight alteration to the active site. It's no longer complementary to the substrate. And that's all caused by the inhibitor binding to the allosteric site. When we consider these non-competitive inhibitors, it's worth remembering that they can bind either reversibly or irreversibly. It depends on which inhibitor we're talking about in which enzyme. If we now consider that same graph that we looked at with our competitive inhibitor, substrate concentration on the x-axis, rate on the y-axis. We've got our red line, which is showing the rate of our reaction with no inhibitor, and then we've got this blue line, the lower line, which is that same reaction, but we've added a non-competitive inhibitor. The key difference between these is, unlike our competitive inhibitor, where they both reach that same maximum rate of reaction, the non-competitive inhibitors do not reach that same maximum rate of reaction. They in fact lower the maximum rate possible. What we also find is that by increasing the substrate concentration, you can see we're not able to return that rate of reaction to the same level as it was without the inhibitor, because where they're not competing for the same space on the enzyme, the active site, then increasing the substrate concentration isn't going to alter your rate, because obviously it doesn't have an impact as to whether it can bind or not in that region. The last aspect of inhibitors we're going to look at in this video is something called end product inhibition. It's quite self-explanatory for the name as to what this is. Basically, what we mean by end product inhibition is that the final product of our reaction is going to remain bound to the enzyme, which then prevents further catalysis taking place. So what we're really looking at here is an example of a negative feedback system. Let's look at this in a bit more detail then. If we think about a range of metabolic processes in our living organisms, then what we tend to find is that these are going to be a series of enzyme catalyzed reactions. So it's not just a single reaction catalyzed by a single enzyme, but we actually have this little series of different enzymes catalyzing different reactions as they go through the larger process. And a great example that's going to come up in one of the later units is respiration. What we then need to do with this end product inhibition is basically create a system that's going to stop that end product accumulating in too large a quantity. So what we actually find is that the product of that last enzyme is going to act as this non-competitive inhibitor for the first enzyme in the whole process. So if we have a little look at our diagram at the bottom, then what we can see, first of all, we've got our first enzyme and our first substrate. So obviously the active site of enzyme 1 is complementary to substrate A, it binds, and we release the chemical B, our first product. Now B has got the active site that is complementary to it on enzyme 2. So again, that one binds, it's going to undergo its reaction, and we make product C. Product C has a complementary active site on enzyme 3, goes through its reaction, releases product D, and then D has the active site complementary to it on enzyme 4, and that is going to release our product E. Now, E is the one that's going to act as our end product inhibitor. E is then going to bind to an allosteric site on enzyme 1 again, And what's going to happen here is it's going to change the shape of our active site. So because we've changed the shape of the active site on that first enzyme again, it's no longer complementary to substrate A. So we're going to bring this whole process to a stop. Because if we're obviously not able to bind substrate A into the active site, that means we can't make B and the rest of the process can't happen. It is worth remembering that obviously when the concentration of our end product then falls, as it will when obviously those products are used up in other processes, then we will release it from the allosteric site, and therefore it's then going to allow the active site to return to its original shape. A can then bind to it and the process restarts. The final thing to bear in mind when we're talking about our enzymes and these enzyme reactions is when we're talking about these metabolic processes where we've got a whole range of different reactions going on requiring different enzymes, then what we tend to find is all of the enzymes required for a given process are located within the same region or the same organelle within that cell. And this is all about efficiency. You don't want a situation where you have one organelle that's involved in one part of the process, and then you've got to transport the products from that all the way to a completely separate organelle for the next part, and so on. So we tend to have these dedicated organelles or regions that allow us to have everything required for a given process, because that is way more efficient than any other technique. As always, don't forget to subscribe to the channel so that you can see when we update any more videos, and head on over to the A-Level Biology website where you can find a range of other resources that will hopefully help you in your biology studies.